Hello everyone, my name is Andres Medina and the name of my partner is Manuela Maldonado, and today we are going to present our seminar. The topic we choose is the USP7, the ubiquinase, and stabilizes the EZH2 in prostate cancer cells. Okay, the prostate cancer. The prostate cancer is one of the most common type of cancer in the men. The size of the prostate is like a nut and it may grow as the person grows older. The prostate is a gland. The glands have a lot of cellular exchange, so this may cause a mutation and the damage in the future. Also, the prostate helps to produce the seminal fluid, and the exam to check the prostate is called the digital rectal exam, also known as DRE, and consists when the doctor introduces his finger into your rectus to check the size of the prostate and also the other conditions.
Molecular biology is an essential aspect that concerns significant medicine. The area of research and trial in medicine is the core for future advances and new discoveries in this field, and they wouldn't be possible without molecular biology. It is a way to understand every little detail from the most small to complex aspects in the human body. Additionally, the article showed how the USP7 promotes invasion cell migration and sphere formation, partially through the stabilization of the EZH2. Lastly, the article results demonstrates the inhibition of the cell migration, proliferation, invasion, and sphere formation abilities by the combined therapy of the P5091 and the EC. Okay, so the objective of this article was to evaluate how USP7 ubiquitinates and stabilizes EZH2 in prostate cancer cells.
Okay, the methods used in this experiment were immunoblotting. This technique, also known as Western blot, is used in the molecular biology to identify a specific protein in a big amount of proteins. The proteins were separated by sodium dodecyl sulfate polyacrylate, held electrophoresis, and transferred to membrane and filter papers for Western blotting. The knockdown method is a method used for studying a specific gene. This method consists by eliminating the gene from the experiment. When the gene is eliminated, you can actually see how this may vary and you can discover the real functions of the gene. In this case, they introduced two small strained interfering RNAs into the cytoplasm to produce later retrovirus.
Another technique used in this process was the RT-PCR. Its foundation consisted on developing an amplification of specific sequences of genetic material. In this case, they made an amplification of RNA messenger in which the expression of a gene was evaluated. The gene that has been evaluated was the GAPDH for normalization. This was in order to show their intervention and relation with prostate cancer. Additionally, the in vivo ubiquitination was another technique used. This one was performed on a living tissue or organism, searching for their objectives, which was the relationship between the USP7 and the EZH2 involved in prostate cancer, and how the USP7 will potentiate the EZH2, generating a negative result such as cancer.
In the first graphic, there was an endogenous interaction between the USP7 and the EZH2. Also, the U145 cells lysates were immunoprecipitated with normal IgG or anti-EZH2 antibody and immunoblotted with anti-USP7 or anti-EZH2 antibody. In the second graphic, we can identify the consistency of the targets. The targets were tested with the USP7 and the EZH2. Also, the black lines showed in the graphic determine the consistency in every reaction. In the graphic E, as we can see, every sample reacted with the EZH2. This supports the theory proposed by Shaw at 2019. Also, the immunoprecipitation assay showed that the USP7 of fully linked the similar domain to UBI12 and the domain UB345 in the domain C, they interacted with the EZH2. Also, some proteins like GMPS, ICP0, FOX04, and the NMT1 interacted with the CTD and the USP7. In the graphic A, every target is positive. So the B-actina and the HA-EZH2 reacted very similar. So there isn't a very notorious difference in this reaction. Okay, for this graphic, they transfected HEK 293 T cells with ARN HC of USP and mutants EZH2 marked with HA. In contrast, USP7 knockdown and P5091 treatment reducing the protein's levels of all the EZH2-GOF mutant cells. So, the cells were lysated and immunoblotted with anti-HA or anti-USP7 antibody. And for the last graphic, the graphic F, they transfected HEK293T cells with flax USP7 and mutants EZH2 resistant to therapy with HA. The lysate cells were submitted to immunotransference with anti-HA antibodies or anti-flag. So this, like in the past graphic, showed us that there was a overexpression of the USP7.
In figure 3 shows how the USP stabilizes oncogenic EZH2 mutants. In the graph E, they are comparing lysate and IP, alpha HA. So, in the HA EZH2, there is no significant differences between both tensions. The cell lysates were subjected to immunoprecipitation with antigens and antibodies. The two therapy-resistant EZH2 mutants were found to interact with the USP7. Okay, so now in graph H, they use the beta-actin, which is a test control, and it is a protein present in all fluids. As the GOF EZH2 mutants, the protein levels of their spirit-resistant EZH2 mutants were increased by the USP7 overexpression. And in graph H, whereas the USP7 knockdown and P5091 treatment destabilizes the therapy-resistant EZH2 mutants. Now in graph A, we see the negatives and positives of each element being labeled. There are no big differences or alarming results. Thus, USP7 is implicated in the stabilization of wild-type and oncogenic EZH2 mutant proteins. These results suggest that USP7 specific inhibitors could be promising therapeutic agents for the treatment EZH2 dependent malignancies.
Okay, in the discussion, the reference we chose were, I'm gonna read. The ubiquitin proteasome system (UPS) is involved in many biological processes, including cell cycle progression, signal transduction, immune response, stress response, and tumorigenesis. This was true. The second one, a Naser offset homolog 2 EZH2, a member of polycomb repressive complex 2, PRC2, trimethylates lysine 27 of histone H3, H3K27, Me3, and regulates several genes involved in tumor development. That was true too. And the last one, mutant forms and abnormal expression patterns of EZH2 have been detected in several cancers and are frequently associated with tumor progression and poor prognosis. And this last one was also true and was corroborated after reading the text.
Okay, so finally, the conclusions. Molecular biology is an essential aspect that concerns significant medicine. The area of research and trial in medicine is the core for future advances and new discoveries in this field, and they wouldn't be possible without molecular biology. It is a way to understand every little detail from the most small to complex aspects in the human body. Additionally, the article showed how the USP7 promotes invasion cell migration and sphere formation, partially through the stabilization of the EZH2. Lastly, the article results demonstrates the inhibition of the cell migration, proliferation, invasion, and sphere formation abilities by the combined therapy of the P5091 and the EZH2 inhibitor.